Okay, good morning, everyone. It's my pleasure to introduce Dr. Marnin Wolfe, an assistant professor at Auburn University. Marnin Wolfe got his PhD from the University of Pittsburgh in ecology and evolution. Then he transitioned to plant breeding and quantitative genetics for his postdoc, what he did at Cornell University under the supervision of Jean-Luc Ménink. Dr. Wolfe worked a lot on cassava breeding at that time through the Next Gen Cassava project, developing genomic selection, multivariate prediction, mate selection, and so on. Since 2022, Dr. Wolfe has been appointed an assistant professor at Auburn University, working on quantitative genetics, and he is now breeding for intercropping on cover crops. So today, Dr. Wolfe will be talking about how to use genomic selection for intercropping. Please welcome me, Dr. Wolfe.
Well, good morning, everyone. First of all, thank you, Charlotte, and also Albert. You guys have been awesome hosts, and to all of you for coming. I've had a great time getting to meet folks yesterday and also this morning. So it's just been great. And so I hope I have some interesting stuff to share with you. I decided on a relatively bold title. Can we use genomic selection to breed for intercropping? Sure. It's a well understood tool that if you throw enough money at it, you probably do. So what I really want to talk about is our early efforts to attempt to do some breeding for intercropping that in some cases are using genomic selection. So I'm gonna break the talk up into like three parts. And first we'll talk some of the idea, what we want to do to improve intercropping. I gotta kind of tell you about what I've really been doing the last 2 years, which is establishing a breeding program essentially from scratch. It's focused on cover crops and forages at Auburn. And then I've got a couple of different breeding for intercropping projects that I have hopefully enough time to share enough detail with you. But this are really early days. Don't expect like genomic prediction accuracies and answers to your questions. Just plans and projects we're getting going. All right, so actually I think Charlotte really covered this very well. I started as an evolutionary ecologist, spent 9 or 10 years doing genomic selection in cassava, and kind of putting together my life as an ecologist and as a quantitative geneticist together. And that's kind of why my focus is on adding sustainability in our cropping systems, mainly with cover crops and breeding for intercropping. Okay, so let's talk about just like the idea, what we want to do and why. As I was doing my own faculty search and trying to like chart a course into this area, I wrote a perspective article, just kind of waving my hands about what I think we could do, along with Jean-Luc Ménink, Mikey Cantar, and San Antonio. And also it was part of a review article that was published a couple of years ago, senior authored by Aaron Lorenz, led by Ginny Moore. So we talk about a number of the intercropping systems that we think could benefit from breeding that are relevant in temperate systems. So some of those ideas are going to come out in this first section here. But of course, first thing is what am I talking about? Intercropping is any time, according to me, that you grow 2 or more species and or cultivars in the same place and or at the same time. So I'm using it as a very broad umbrella term, and indeed it's a really ancient practice with lots of different synonyms like polycultures, different ways people like to call it. So very diverse set of options. In fact, here's just a myriad of different configurations that could fall in this category. Just for time, I'm not really going to go over all of them, but mostly I'll talk about mixtures today, which just means we're planting multiple species together, potentially harvesting them together. So mixing the seed, putting them in the same hopper, they're in the same row together. Obviously, there are a myriad of other options. You can kind of break it down into lots of variations on spatial and temporal segregation of the crops. So there's some time and or space period where there's interactions between crops, right? So I'll just give examples of the systems as we go. So I'm mostly focused on mixtures because, folks actually sell bags of cover crop mixtures or forage mixtures at a bag of seed that doesn't really have breeding under the hood, right? So All right, so I do need to kind of justify this. I could probably spend a whole lecture talking about why to intercrop and under what circumstances. From my conversations last day, it seems like we seem to be talking like everything should be intercropped and then sustainability will occur. That's of course not like what we're going for. So I'm just going to give you two general like overarching benefits that could occur from intercropping. And the first is they could yield better when they're optimal, when they're done right. So this is like illustrating a seeding replacement rate between two species, so the monocultures are there. And just the concept is that the mixture does better than the monoculture. If you're familiar with the term, there's also the term land equivalent ratio, which means that it means the same thing. And the other, much less easy to directly quantify or say what I mean, is often called multifunctionality, which is just like more species, more features, basically. It's conceptual only, of course. We want more out of our cropping system than just the yield. So I think that's pretty well illustrated here. So for a cover crop example, rye as a grass produces a lot of biomass, so it can contribute to soil organic matter and has allelopathic properties, can help with weed control. Pea is a legume, so it helps to fix nitrogen and help potentially with the yield of the subsequent crop. So just the idea is, if you have more diversity in your field, you can get some of both of the benefits when done optimally. So also, before I talk about how we're going to do this, I just do want to highlight that to realize the benefits of intercropping, it involves a lot more than just quantitative genetics and genomic selection, right? So I'm focused down here on breeding scheme design, but as this is showing, like we need several what they're calling enabling factors to support this, right? There are certain policies that would be beneficial, including like funding for our breeding efforts would be lovely. Markets need to support this in various ways. And of course, cropping system research is really, really vital. I'm relying on agronomists to tell me what seeding rates and planting dates and those kinds of things so that I can focus on genetic evaluations. Okay, so in our review article, and this is more complex than I'm going to actually walk you through, we kind of came up with a process map, if you will, that just talks you through how you might think about designing a breeding scheme. So to make it faster, I just kind of simplified this my own way. So first of all, we'll just assume the enabling factors are there. We've got a good motivation to do this. We've got, let's assume, funding to do this. And a good part of that is let's say we already know we have variation in performance in intercrop across varieties in one or both of the species that we're going to work on. Otherwise, we would just be doing agronomic research, trying to improve management. So in that case, we're going to need to ask questions about like how variation is expressed. So obviously, if there's no interaction at all, there's no need to breed for intercropping. This is basically like a G by E kind of question, right? So if the interaction between monoculture and intercropping is non-crossover kind of interaction, you don't get re-rankings, you can potentially just keep your, like most of our breeding programs for monocultures already, right? And for the most part, the most feasible thing would be to not alter that and start doing all these new trial series. So if this is the case, then maybe you can just breed for intercropping in monoculture. For example, we can also maybe find proxy traits. They're expressed properly in monoculture. We can use those that are predictive of performance in intercropping. Just keep our breeding pipeline as it is. But obviously, I'm thinking that we're going to see that there are crossover interactions. So intercropping represents a different environment, and in some cases, at least, the best varieties in an intercrop won't be the best in the monocrop. So in that case, you definitely need to breed for intercropping with evaluation under intercropping conditions, right? So this is probably could have been better illustrated, but like let's say there's a possible spectrum of different co-testing strategies that we could choose. The easiest or like maybe the cheapest would be to keep separate evaluation of germplasms and use, I could have illustrated this better. We could use a tester scheme, similar to how we do in hybrid breeding. You have your germplasm of one species and you're gonna test it against one or two or a couple of cultivars of the other species. On the other end of this co-testing spectrum, we're gonna like join two breeding programs together and come up with a trial series that, you know, to some degree co-tests the early stage germplasm, the late stage germplasm. So we have to pick something that makes sense for any particular system that's, you know, along the spectrum of options. Or in other words, we're not always going to do the same amount of joint evaluation of germplasm, but when we do, this is what I want to talk about, how we are going to potentially do it. And actually, I don't have to invent very much. The ideas have been around for a while. It's a paper from 1985 that I think first described the basic kind of scheme that you need to use. So here they just have two pools, families of species A, families of species B, and just for simplicity, we're just going to assess random pairs of A and B families. We're going to do some version of a co-testing strategy and use those information to do evaluations to decide which we're either going to select some members of family A or species A and or species B for further intercrossing to like improve the populations. So you do some kind of field trial is basically here where you have some intercrops evaluated. And then we also, so we can then genetically evaluate that in a fairly straightforward way, actually. So this is the most straightforward model that we can use, and I don't really have to invent new statistical machinery or anything here. This is often called a general and specific mixing ability model, where a response variable is the joint yields or the joint merit of the intercrop. There are other ways to do this where you take separated yields, and I may have some time to talk about that at the end. But so in this case, we have the total yield of our intercrop, and there's main effect for, obviously this is an oat and pea example. There's main effect for oats. So this is the average performance of each oat genotype or entry against the average pea background. We have a main effect for pea, that's the same thing. Average effect or average merit of a pea in the background of any oat, and potentially some interaction effects. That's the deviation of performance. We call that specific mixing ability between the two pairs of genotypes. So this is basically the evaluation scheme. There's some variations on this that we can do, and it has direct analogy to general and specific combining ability from hybrid breeding. So we can kind of use a lot of those ideas, and indeed that's what we're doing. So genetic evaluation is, of course, what we need to do. The challenge with fitting those simple seeming models is talking about breeding and genetics, agronomy with more than one species. So the number of combinations and interactions that we might have to navigate, sources of variation are almost daunting, right? Genotype by species, by environment, by management interactions, they're all going to be there, potentially. So just by example, on the genetic sides, it's pretty easy to imagine two breeding populations with 200 individuals each. That's 40,000 pairwise combos. We're never going to field test all of those. We're definitely not going to do replicated multi-locational field trials with all of those. So we need alternative strategies. So obviously, conventional phenotypes only selection means that we have to evaluate all of those combos, or at least most of them, depending on whether we want the interaction term. So we're usually going to do this, and if you look in the literature, the couple times where these sort of parameters have been estimated, they're estimated with small populations that we could say are late stage breeding lines. So you don't have a lot of genetic variation to still select from. So just like we want to breed evaluation for intercrops and whatever we can as early into the breeding pipeline as possible to get good results downstream. So one thing we can do to start to get at this more attractively that doesn't involve any necessary genomics or phenomics is just sparse designs. So sparse designs are very important already in the setup of genomic selection training populations, especially when we're trying to capture genotype environment interaction. So for intercrops, what I'm talking about is we don't have to evaluate all combos of both germplasms, right? Especially if all we want is that main effect, the general mixing ability component. So this is a study from 2021 that had 28 peas and 7 or 8 barleys. And basically, so that's 196 combos. I'm sure they had a couple of replicates of each, but they only tested 64 combos. So every pea had 2 barley, every barley 8 peas approximately. And from this, you can get a decent estimate of that main effect term, and that's useful for selection, no doubt. So this next is not a very good illustration of the idea, but all I want to do is put a genomic relationship matrix on it, essentially. That is, we can test even more environments. In this case, I'm talking about intercrop partners by using genomic relationships, plus these sparse designs to connect better all the different field plots. Okay, so that is basically the idea from the perspective article we wrote a couple of years ago, as let's do joint genomic selection for two populations. So in this extreme co-testing example, what's illustrated here, I'll just direct you to some of the key parts. This is like meant to be a grid of all possible combinations of the purple species and the red species. Okay? So instead of testing that intractable number of combos, we'll have a sparse design. So you should see there's a filled tile, meaning a combo we are going to put in the field for every member of every species, but we're not testing all with all. We're testing all with some, right? So there's some variation in partners for every entry in both species. Now we get the main effect term. And the other part of this, so 3 and 4 here, let's just put it out of the field. We'll run the appropriate field trial or whatever the intercrop is, planted, harvested together, and measure whatever is relevant, biomass, yield, etc., and develop a genotype everything. And so in the end, we're aiming to predict the benefits of intercrops for all genotypic combinations. So the general and specific mixing ability components I was just talking about. So with those predictions, hypothetically, we can then do at least 2 kinds of selections. So first kind of selection would be just advancement to a next stage of trial. So the statistical machinery says that I can predict specific combinations. So even if I have not field tested them, and so I can perhaps have a field trial that we're gonna actually try to validate that. We're gonna plant out all the combos of that were predicted to be best. The other kind of selection, this is a little more complicated looking, but it means just recurrent selection for population improvement, advancing parents. So this is depicting like a field trial sequence, simplified, of course. So like trial 1, advance to trial 2, advance to trial 3, multiple generations are going on. And so at the end of the field trial, or at some point, we're advancing better parents for crossing. So we've got 2 gene pools, and this is very idealistic, no doubt about it. But we're talking about getting this ultimate outcome, right? We're going to co-adapt 2 germplasms to each other and get, cropping system-wide genetic. So yeah, again, this is like the concept, idealistic, no doubt about it. And this is just meant to show that the statistical machinery is there. So here's your general and specific mixing ability model again. These are basically random effects for the main effects for oats and peas, respectively. And we can basically just genotype them and connect the plots with genomic relationships for oats, genomic relationships for peas, and at least the most straightforward first way to predict the genome by genome combinations is just a chronicle product of the 2 matrices. So this is really just to show that the statistical machinery allows me to do this. Whether or not it is accurately predictable or not is a very big and important question. So what I want to do is start generating some empirical data. So okay, those are the concepts. This is kind of the big idea. I started in Auburn a couple years ago, and we're establishing a breeding program that hopefully, among other things, will let me test some of these things. So I am down here just to the left of Atlanta in central eastern Alabama. We grow cotton, peanuts, soybeans, and corn are the top crops, not particularly in that order. Other things to say, I kind of picked up on forage breeding legacy at Auburn. As I was introduced, I'm assistant professor of quantitative genetics, so I can kind of focus on the cropping system that I can fund, basically. And so there was germplasm since like a breeder retired in 2016, and what I'm getting started with is largely what I could revive from his cold storage. Right, I guess I already said this part. I'm chosen to support southeastern growers to benefit from covers from forages, and breeding for intercropping is the sort of major research focus. Very practically, the real thing that I've done the last couple of years is I started to build a team, and I think this is the most tangible thing I have to offer so far. We're 6 as of now, including myself, not including a couple undergrads who work in the lab. So Gideon Oyebode is first PhD student to join from Nigeria. Philip Castillo is from Philippines. He started with me just like 6 months after Gideon in spring of 23, as did Francis Manze, is doing a PhD. He's from Uganda. And just this summer, Emily Takashita became the first Alabamian student to join the lab. And Cynthia Gogo becomes the first postdoc. She just joined a couple weeks or beginning of September. So yeah, we're a small group, but we're growing. And also starting a breeding program. I was asked a bunch of questions for grad students about like transitioning between crops and starting a breeding program. How do you do it? You ask a lot of questions, you depend on a lot of people. This isn't any particular order. And for time, I'm not going to try to list people, but just to say I'm very grateful and depend on lots of people. So team efforts. All right, let's briefly talk about cover crops. I somewhat assume y'all are very familiar. Cover crops are planted to benefit the soil and subsequent crops rather than for harvesting for profit in general. So they are going in when you're harvesting your corn or your soybeans, and they're coming out before they're being terminated or tilled under right before you're replanting. And I could spend a whole lecture on cover crop benefits, and of course it depends on the cover crop and the region and the grower and the crop that they're growing. So let's just say there are generally various kinds of things we call ecosystem services, like improving the organic matter of soil over time, of course, increasing yields of the subsequent crop, fixing nitrogen, those kinds of things. There are also economic incentives, subsidy programs, namely. So EQIP is a NRCS program. And so you can see here in Alabama, a farmer can get $50 an acre for planting a single species cover, and hint, they can get a small premium for planting cover crop mixture. So these are kinds of incentives for doing this kind of work. And indeed, cover crop acres are increasing year on year. So there's a trend down in the southeast in Alabama. We're actually above the national average for cover cropping. So it's the right practice down there as well. And I'm not alone. I'm actually joining up with the Cover Crop Breeding Network, which is a national and well-established team that features Nancy Elke here in Minnesota. So I'm really just adding Alabama. So far to the list, we've participated in the advanced line trial for crimson clover last year. And actually, as of now, I will be the lead crimson clover breeder for the network. So I'm going to be much more involved in the future, which is really exciting to me. Yeah, so can't talk about all the details or all the species, but essentially there's 3 legume crops, crimson clovers, hairy vetch, and winter peas that are focused on in the network. Cereal rye and brassicas have been added recently. Also won't spend too much time on traits, but essentially we look for biomass, for early vigor, we look for winter survival, and some various other things depending on the species. So for my case, of course, I say we need to select for performance in mixtures. This is actually this pie chart's from a SARE survey of cover croppers and shows that 65% of them, the green here, are already planting mixtures when they plant cover crops, and they plan to continue to do so. So that's motivation for this kind of work. And this is kind of a transition for me to talk about crimson clovers, which are, I'm not going to talk about all the species we're working on today. Crimson clover is kind of the lead species I'm trying to actually do some breeding work with. It performs really well in Alabama. It's actually, like, according to surveys, it's the lead legume cover crop in North America, although you're not going to see much of it around here. It doesn't quite like, maybe North Carolina, it starts to get winter killed too much, but loves it where I live. So a couple details, it's a cool season allogamous annual, which means we're breeding varieties that are open pollinated populations. There is some self-compatibility in the species, and it's really lovely and also smells nice. We don't know much about the genome. That's obviously going to be an important thing that we are working on. I'll talk about it only in the very briefest sense We think it's a diploid with a 2n of 14. We think it's got a 600 fit. This is based on like C values. So we're working to develop the genomic resources for the species in parallel to the other work that we're doing with it. And as I mentioned, I started with Auburn germplasm. So there's a couple of popular crimson clover varieties that have come out. I've got breeder seed. So this is really one of the first things that I've been, got some traction on. Okay, so very quickly, we are doing trifolium resource, genomic resource development. I don't have time to talk about this, but we are basically developing some de novo reference genomes, do some resequencing for collection using some wild relatives. So crimson clover is certainly there. We can talk about that if you have questions. So at this point, my master student Philip has figured out how to clone crimson clover, immortalize a line. We've submitted samples, and so a reference genome or sequences should be on the way. Hopefully sometime next year we have something we can start working with. Okay, so a little bit about how we breed crimson, because this is going to feature how I'm actually trying to do evaluation for mixtures. Said it's open pollinated, so we start with space plants, and every plant is its own special individual. So we'll call them F1s, no inbred lines, remember. So basically, we do some culling selection in the field. We plant a nursery in isolation from other clovers. We're going to maybe allow the top 10 or 20% to flower, get a couple hundred plants that give seed, and then we're going to collect seed from individual plants. So they've been open pollinated. We know the maternal line, the maternal lineage of each entry, basically. So we use the seed that comes from these in several different important ways. Firstly, they go into another space plant nursery the next year, next round of selection, and the continuation of the lineage. The other thing that I want to do that's not actually part of the sort of standard breeding pipeline is an early stage evaluation. Like the next year, we take those half-sieve entries and we put them in what are often called sword plots, because biomass yield is our top thing. You need multiple plants for that, because if I'm waiting for seed, these guys senesce, so you can't get biomass from them. So each plot here is a half-sieve, or in some cases, a bulk line. So that's one of the features of the genetical units that we're using. The other thing that we do with the selected plants from space plant nursery is create a bulk, also called a synthetic line. From the selected parents, we combine equal portions of seed to basically make an advanced population, if you will. Those go into the Cover Crop Breeding Network's advanced line trial, which is a multi-year, multi-location trial. This is the first time that we get yield data in terms of biomass and quality from any given entry. There's also going to seed increase as well. Okay, I think I have time to talk about our actual projects that are trying to work on intercrops. So I was very fortunate in my first year to get a couple of, to get some funding behind a project that's using crimson clover mixed with oats as a cover crop mixture, a focal cover crop mixture. We picked oats because it actually has better forage quality than the most common grass, which is rye for cover cropping. And there's a bunch of folks down in Auburn who are looking at grazing cover crops. So this is the focal system we chose. I have an Auburn internal grant, it's like $50K 2 years, and a NIFA seed grant that's also 2 years, $300K. And that has allowed us to expand to a 2-year, 2-location pilot trial. And that's basically what's underway right now. So my collaborator is Esteban Rios, who's forage breeder and geneticist at the University of Florida. That's my second location. And I also have Alvaro Sanchez in Val, who are physiologists and like a phenomics specialist at Auburn. Right, so just for the maximum simplicity, our main objective with this pilot experiment is just to demonstrate the sparse design, genomic connectedness to predict mixability at early stages, like I've just described to you. So, so here's basically the idea. We start with our space plants. Our space plants get selected, so we end up with half-sieve seed. We're going to genotype these. I'll tell you a little bit about how we do that, or some of the nuances of doing that in a moment. Those half-sieve seed then go to the 2 respective. So we make a bulk for advanced line trials. They go to the next nursery. The other thing that I want to do is then compose a yield and mixtures trial, as I've been calling it. So those half-sieve plots are now mixtures with oats. So I'll explain that a little bit in a second. So basically, we now have these plots where we have two populations co-evaluated. We genotype everyone from both populations, and we have phenotypes and genotypes. We can then do genomic prediction of mixing ability to hopefully allow us to make selections for mixing ability when we choose what we plant in the next nursery. Okay, so the next slide is way too busy. So I'm sorry for all the words. I'll just say a couple critical things. There's all the details about the experimental design. So we'll have 2 locations in 2 years. And maybe I'll just point you down here so we had a plot budget for 1,200 plots per year, 600 per location. So that means we had between mine and Rios's crimson clover populations, we had 178 half-sieves with enough seed for the experiment. We have Steve Harrison and Ali Babar are small grains breeders in the south, and they have donated us 26 oat lines. We're more focused on crimson in this project, but that makes 4,600 possible combos, which is actually not too terrible. Either way, we're sampling about 22% of that space, that combo space. So if that's 1,020 combos and we have 1,200 plots, hopefully you can see that the design is very sparse with respect to those specific combos. Most of them are unreplicated. However, each crimson clover accession gets plenty of plots observed in both locations. Each oat accession, because there's only 26 of them, they get plenty of clover partners that they get observed with. So we may not be very able to capture the specific mixing ability component, but we should be able to get at the general component, the main effects. This is just a picture of the field trial, because I can show that. We just finished we're actually just next week, we're going to start planting we're going to plant the second year of this field trial. We had only 6-foot plots, single rows like this in the first year. In the second, we have much better seed yield, so we're actually going to have 12-foot plots, which will allow us to do additional biomass harvests. Just going to skip ahead a little. I can say we did a lot of phenotyping. As much non-destructive phenotyping as we could, things like canopy height by species, canopy cover imaging by species, used multispectral and hyperspectral drones. Our final at the cover crop termination time about mid-April, we take biomass, we separate by species, dry them down, so we have separated yields of the two. Okay, so we're doing a lot of phenotyping. That's what I could just summarize that as. Also to mention, so I explained crimson clovers are open pollinated. So one of the other things that we're trying to develop in parallel to this, our field plots are half-sieve families. So every individual is different, but they're all maternally related. So we have different genetic units. We have maternal half-sieve families, and we have balanced bulks that might be in yield plots. So the strategy we're going to use is basically to do sequencing to capture allele frequencies and co-frequencies between families rather than genotypes, because they're not genotypes. So far we have done some low-pass sequencing by Hudson Alpha Institute, and I don't have results to share with you yet. We got our sequences back September. Postdoc Cynthia started September, so she's working to extract. So, from the sequencing at Hudson Alpha, we get an imputed data product, but it's assuming that we have diploid genotypes, so it's really not usable for our purpose. So we are pretty close to actually getting the matrix that we need to start doing genomic predictions here. Okay, so I have some time at this point. If anybody has questions, I suppose y'all should interrupt me and ask. But I also want to talk about 2 other projects in different, well, for just a moment. So I used to work on cassava, as did my PhD student, Gideon Oibode. So cassava, I didn't actually introduce the crop. It's a root crop. It is staple throughout the tropics of the world. About 500 million or more people depend on it for calories every day. So Cassava is also commonly intercropped with many different things because it's a food security crop. So there's a big class of growers who are just growing in their small plots in their backyard and using cultural practices that I know very little about. So one thing cassava is intercropped with is cowpeas. Cowpeas, also called black-eyed peas, Vigna unguiculata, is a short season crop. So cassava grows for about 12 months, cowpea 3 to 4. So they have pretty different seasons or season lengths, but you can plant them together. So Gideon came to me from a cowpea breeding unit that he was working on intercropping with millet and had previously worked with the cassava unit. So we got to talking to Dr. Ismail Rabi, who's the senior cassava breeder at IITA, and we're able to convince him to plant a pilot trial for us. So we picked 120 cassava that came from their PYT and AYT, so like mid-stage breeding lines. We're using a tester scheme in this one, so we're not doing our big sparse designs kind of thing. We picked 2 cowpeas. One is an upright type that's short season, and one is a slower spreading variety. We planted this trial with both mixture and monoculture, so we are able to look at the interaction between the two. And this is basically our plot configuration. So we had cassava stems mixed with cowpea directly within each plot. So far, I have been unable to get very good pictures of this field trial from my collaborators. So I'm really hoping to do better this next year. But so we planted September, cowpea 23, cowpeas harvested December 23, and we've cassava not until August, right? So big amount of time between harvesting cowpea and then harvesting cassava. My bet was against very much impact on the cassava. And we're doing the second year of this trial. Hopefully this will be pilot data we can justify expanding the work. So Gideon has produced some results on this first year of the trial. So this is showing root number for cassava and 100 seed weights, I guess, for cowpeas. Basically, it shows us that cassava that are monocropped do better than the cassava that are intercropped. This interaction is not significant between the varieties. And same thing for the cowpeas. When they're monocropped, they do better when they're intercropped. But nobody should be too terribly surprised by this. I guess I was not expecting. There's a reasonable chance of reason to believe that 6 months without the cowpea would be enough to recover, but that wasn't the case. So actually, I haven't dug into this yet, but one interesting thing is heritabilities are a little bit elevated for many cassava traits relative to monoculture when they're intercropped and when they're monocropped. But I need to look at what's actually going on under the hood here. I'm just skipping over some stuff for time. So another way we can express the difference between monoculture and intercrop in the cassava system is what's called land equivalent ratio, just the ratio of yield and intercrop to monocrop. So a ratio greater than 1 means it does better when monocropped or intercropped than when monocropped. So in the green here, each of these bars that you can't see the names are different cassava lines. And so you can see we have a bunch of cassava, or we have some cassava lines that have an LER greater than 1. So they actually did better when they were intercropped than monocropped. And also, I'm not showing like mixed model results here, but there is a very significant genotype by management interaction. So there's crossover interactions going on. Cassava that are intercropped, the best intercropped cassava are not the same best monocropped cassava. So this is justification to do some evaluation of some kind if we want to select the best for intercrop. Okay, very briefly, I have another project that will get started in the spring that focuses on grain intercropping. So this is an oat and pea project. So oats and peas are a really convenient model compared to like crimson clovers that are open pollinated. Both of these species are inbred. They've got genomic resources, not a tremendous number of different pea breeding programs, but one of the nice things here is if you get the timing of the crop right, you can plant it together, you can combine it together, and then you can separate the seed because they're pretty different in size and density. So this is in some ways a convenient system to use as a model for studying how we might breed for intercropping. And I have a really awesome team behind me to do this project. We got a Coordinated Innovation Network Award from IFA this year. It's almost $1 million for 3 years. So really excited about this project, but won't start till the spring, so I don't have any results to share you. And I have a really awesome team. Jean-Luc Yannick leads from Cornell. Juan David Arbelayas is an oat breeder at the University of Illinois. So his oat germplasm will contribute to our project. We also, of course, about location. NDSU is currently without a pea breeder, but they have a big pre-breeding program. So Marisol Berti has taken over for Nonoy Bandillo. She's a specialist in intercrops, so that's really quite beneficial for us. And we have public-private partnerships. So Sarah Lear and Frank Techno are both Corteva scientists who are passionate about this kind of stuff. And so they're also running a location of the trial. So just for we have 5 locations, and the main goal here is just to pilot genomic prediction for intercroppability, doing some phenomics tools development. I'll just share you a little bit about the design that we're going to use. So we're taking the breeding germplasm directly from mid-stage breeding lines. So we don't want to do like diversity collections. We want to integrate breeding germplasm directly. So we have 480 oats approximately that we expect from Arbelayas's program, perhaps 440 peas. And what we're budgeting for is to do 400 plots each over 2 years. So that amounts to 10 site years, or about 4,000 total plots is what we're going to manage. So with 440 by 480, that's 211,000 combos. So a lot bigger possible combinatorial space when you look at what we're dealing with 2 actual breeding programs. So we're sampling less than 2% of the space. So obviously most of our combos will be unreplicated. Okay, actually I will just conclude with that. So breeding for intercropping is obviously a big combinatorial challenge. Hopefully we can use genomic prediction and sparse testing to offer like a solution where we can actually make selections for these kind of parameters in early breeding stages. I think the real thing we need is to start trying to do this. So we need empirical examples. That's what I'm trying to do at Auburn. I talked about cover crop mixtures as one of the key focal areas. I'm increasingly interested in the cassava cowpea and other tropical intercropping systems, because they're already showing us that we need to actually address this. And I'm pretty excited about the oat pea project because it's a pretty cool model system. We've got good funds behind us. So, all right, thank you guys so much for listening. I'm sorry it did go way more than 30 minutes. Go figure. So I hope we have time for plenty of questions. I'm all ears.
Go ahead.
Nice talk. I really enjoyed that. So do you, I mean, based on your own data or any of the historical data you looked at, you know what the relative importance of general mixing ability is versus specific mixing ability? Is that being quantified at all?
There aren't too very many examples, and I haven't done like a comprehensive analysis, but like you might imagine, most of the time, a couple examples I can think of, the specific mixing ability term is not very significant. Most of these studies are going to be like fixed effects, couple versus a couple, and you know it's the interaction term, so it's harder to quantify. And I think that's definitely going to be the case, like in the clover notes, for example. And most of us probably would prefer that at breeders, right?
Yeah, sure.
But I think there would be, we would probably prefer that, but you might envision like company could sell co-bred varieties in a bag together. I don't think much right now. Some companies sell cover crop mixture seed, but there's very little like science that goes into what they're mixing.
So the paper from 1984 is 40 years ago that had a diagram for the breeding program. It looks a lot like your cyclical reverse selection program. So it's, you know, we have a lot of examples out there where cyclical reverse selection has gone on for many, many decades. Has anybody done anything like that? Or is it just so far diagrams? Has anybody actually put that into practice in the past? That goes back to 1984.
Not to my knowledge. I don't think so. I'm not remembering off the top of my head what they were actually focused on in that study. So I should probably check that out.
I was really interested in hearing the case study about cassava. Yeah, I've also heard other talks when they said that cassava is a kind of changed resistant type of crop because of its resistance to drought and warming scenarios. However, I imagine that up here is kind of like a challenge to propagate because you use the excellence to propagate. So how does your propagation for the breeding happen?
I totally messed up in not explaining that this trial was in Nigeria. It's definitely not here in North America. Yeah, so and I got that a lot over the years working with cassava. We're like growing it at Cornell and stuff. Maybe in the greenhouse for physiology studies, but no, the field trials run by IITA, which is the CGIAR in Nigeria, with their germplasm, and we're just kind of, we did the design, and Gideon went over and helped to plant it. Yeah.
But still, can you wait until they produce seed? Or how does that look?
Oh, you're talking about how cassava is propagated? It's
Yeah, because
propagated.
you have several lines also tested for cassava, right?
Yeah, so they are seed bearing pretty readily, so they can be readily outcrossed, but it's propagated by stem cuttings. So if I go back to like how the trial was configured, there's like yeah, so this is meant to be illustrating like cassava stems planted between cowpea seeds. And this was just a yield trial, so we weren't like collecting seeds or making crosses or anything like that so far.
When it comes to these initial breeding trials you are doing in Auburn, obviously you probably have a large list of phenotypic characters you'd like to collect. What are the ones that you're prioritizing with any of these new cover crops? And what are the most valuable to you?
So obviously biomass for sure, but beyond biomass, carbon and nitrogen ratio is an important and relatively easy one to get at for cover crops. Tells you about like the value to soil, to decomposition of the crop. We have some commodity funds that are going to actually let us characterize almost all of our germplasm for nitrogen fixation, which is isotopic analysis and 15 ratios. And since we are a forage breeding lab, we are just getting started developing like NIR equations and using existing NIR equations to get at forage quality traits, which as not a forage expert, I'm still kind of learning what those are, but basically we can use NIR on biomass to extrapolate various kinds of phenotypes that are hard to actually directly measure, that are proxies for how valuable it would be for grazing.
Any early season, all those traits seem to be either late maturity or post-harvest. Any early season traits that you hope or you see any early indications for positive correlations you could maybe use to accelerate selection?
Well, definitely we look at, and we do like a vigor score early stage, which is vigor scores are of modest value usually because they are subjective scores that require an expert. I'm developing my expertise, so my vigor scores are probably not awesome, to be honest, yet. So early stage establishment is like really important because we want ground cover early. But we can kind of use this. I mean, we're using imaging from early on, so we have that kind of data. I'm not thinking of, also we look at like how quickly we get emergence, how much emergence we get. We don't get much winter kill in Alabama, but the rest of the network is really focused on like how much kill you get between fall planting and early spring.
Yeah, so I have a question on like the public perception of adult adoption. What's it like in the Southeast? Are farmers really open to adopting this? What are some of the biggest barriers we see? Because at times here in different regions, we're actually seeing lower adoption of cover crops in more recent years?
So we're something, I mean, I don't remember the numbers, but we are above the average for cover cropped acres. There's plenty of like push from extension agents, and I showed you equip payments. There's subsidies are there, and that certainly helps. 50 to $57 an acre, kind of convincing potentially. So I would say in some ways that kind of confounds the long-term maintenance of this practice. Like, will this continue when the payments shift to something else? Wouldn't say that's quite above my pay grade, but I don't quite know the answer yet. Farmers are enthusiastic, but again, like they're getting payments for it. So, and regarding other intercrops, I haven't really quite started to interface with farmers about their interest in such practices, but they're out there.
And we have an online question. Have you seen or expect to see hybrid vigor in the F1 space plants and half-sib rows? And then what are the implications of selecting F1s in breeding trials when you plan to release a synthetic variety?
Okay, so the space plant nurseries are done by culling selection and then open pollination. So I don't even know that I would have a way to say that it's hybrid vigor because I don't know who the parents were. So I can't like compare these two parents made this. Not to say that direct crosses can't be done, and I would certainly like to try that at some point. Clover flowers are very small. So the other part of the question was?
I think they have a misunderstanding of seed bulking and variety releases. What are the implications of selecting F1s when you intend to release a synthetic variety?
Well, so what we're doing is we're selecting F1s or just plants, and then we're doing two things. We do some further testing. So I have things like seed yield, and potentially in the future I'll have like height or size estimates of plants that I can use to. So we do culling, then we have seed, and we can do a little bit of additional selection to comprise a synthetic. I don't know, that's not really answering directly your question. I'm definitely kind of still learning. And one of the things we're doing with all the genotyping, I'm hoping is to start to actually get an idea of what are the genetical units. Like what is a half-sib, how related are all these half-sibs? How much inbreeding versus outcrossing is there? How diverse are these synthetic lines? How diverse are are varieties that are being bulked for actual like sale in the by licensed growers? I don't know those because nobody's looked at that, but it's in some ways you can think about breeding open pollinated crops like this, like you think about breeding animals. It's pyramid shaped rather than a diamond shape, right? So we're increasing the size of the population, hopefully constraining the diversity that ends up on farms. But that's my goal, is to constrain and homogenize the diversity, I guess. Like everything should mature at the same time, look kind of the same.
You had briefly touched on your plan for the genome sequencing and things. Could you elaborate on that a little more in our last 5 minutes?
Sure, yeah. We got the JGI has a program called Community Science Program. If you do anything that's related potentially to biomass or bioenergy, that's their DOE. So they basically provide sequencing resources to develop genomes. Our goal is to develop like a pan-genomic resource. So there, I guess I can actually go back to that. Way back to that. Yeah, okay. So we picked 4 main clover species that have certain different properties, 2 annuals and 2 perennials. So crimson clover is a reseeding annual. Subterranean clover is actually a weird selfing clover that pegs and puts its flowers on the ground. Red clover and Cura are perennials. One is a crown forming, one is the rhizomatous. And so basically we have breeders who have contributed germplasm for each of these species. And what we're doing, so we have a reference genome line being selected for each species. like for the case of red clover and pure clover and sub, they all have examples of reference genomes already. For instance, the new guy. And yeah, so we'll have a lot of data to try to. My main goal is really to try to develop marker sets that we can use applied reading, and so that requires discovering variation. They strongly benefit from having marker order, right? So the genome for that. But this is going to allow us to do all kinds of like population genetics, evolutionary genetics. We've sequenced 49, I think, different additional clovers. We can do a little phylogenetics. So there's a lot that we'll potentially get out of this. But so the JGI gives you gratis access to their sequencing, but they don't support, and there's no like money given. So there's also not like a commitment to a time frame. So they say like six months approximately to a year for results.
For the intercropping between coffee and cassava, have you tried to consider the cost and future in this work? And is it something that's going to be adopted by growers for cassava? Or
my main goal with the trial that we are running so far is to generate some pilot data that might convince a grant agency to fund that work. And if I can convince a grant agency, the breeders will be happy to participate. I don't actually know what the right target is for this because the Gates Foundation and the 1CG sort of thing has shifted the landscape a bit. So I'm just glad that we're able to like put in the pilot, and the evidence so far is kind of in the favor of doing the work. One of the first things I did when I started talking about intercropping was contact our program officer at the Gates Foundation, who said he was very passionate about intercropping, but it would be a challenge to fund because we would have to identify, called it like a mega intercrop, one that's impactful and used across a huge acreage or something like that. So, and in Africa, as I understand it, polycultures are much more common, and there's just a lot of variety in what is grown together. So we're trying to chart a course to do that work.
That reminds me, I was fascinated by your result from intercropping where some of the cassava clones yielded almost 2 times as much under the feed compared to monocrops.
Yeah.
That's remarkable. Do you have any explanation why that would be?
Well, so no, not really. So Gideon gave me that result certainly like shortly before I came over here. One of the things that I didn't unpack in it yet is whether he's expressing the cassava yields only as like, was the yield of the cassava twice? I mean, one way or the other, it's impressive if we had cassava that yielded twice when intercropped and when monocropped. But what you really want to express for the LER is the yield of the total intercrop. So factoring in the cowpea, like if the cowpea took such a hit, maybe the economic merit wasn't there. I'm just not sure which is actually shown there. I want to say something else about that. But, and it's only 2 reps 1 year, so we're doing another year. I think that'll help a lot. So there's a lot of variation in cassava architecture. So some of them are really like straight and non-branching, very tall, and some of them branch out quite a lot. And there are different use cases for the market or for growers for those different varieties. And I think that's going to have a big impact on which cassava play nice with cowpeas. So probably a big branching one is going to be a shade problem for cowpeas.
It's unfortunate that Gates Foundation is interested in that. It's not like interested in just large scale industrial. That's a very good thing. Yeah.
Well, so actually, one of the later things that we've had going on in the breeding program, the cassava breeding programs, is better delineate our product profiles. So now we have market classes. One of them is like for forget what they're calling the market classes, but like conventional growing, big producer. And another is a food security crop. that's more like for a small grower, who's just got it in their backyard essentially. And one of the big differences between those is that architecture. So growers who grow on a large scale want straight stems, and some growers who are small scale, they want like the branchy stuff for weed suppression. So the food security angle, those folks are polyculture, they're intercropping often. So that's the market class that I think we want to target for intercropping. And we've been doing all the breeding under monoculture, not really, we might think about like the architecture and cooking qualities and stuff. We haven't thought about that aspect of the cropping system.
Okay, we're gonna close the session for now, but Marnin will be here for half an hour still to answer any remaining question. Thanks all for attending this, and thanks Marnin Wolfe for this awesome presentation.